And finally, this last section about transcription is going to cover eukaryotic cells. So as you recall, with the bacterial cells, they can go straight from transcription to translation, sometimes even simultaneously. But our eukaryotic cells actually have to modify the RNA after transcription. So when eukaryotic cells produce the product after initial transcription, they produce something known as pre-mRNA, and then they go through the pre-mRNA goes through something known as RNA processing before that RNA can get sent to the cytoplasm. And so during RNA processing, both ends of the pre-mRNA primary transcript are altered, and there's also some cutting of the molecule that occurs. So we'll talk about both of those things. The 5' end of the pre-mRNA receives something known as a 5' cap, and this is usually made up of G nucleotides. And then the 3' end usually adds about 50 to 250 A adenine nucleotides, and this is something known as the poly A tail. Remember, poly means many, so it's just like many adenines. Okay, and there's several reasons why these two modifications are done. One is they help export the mature mRNA from the nucleus to the cytoplasm. So they help with actually getting that mRNA out to the cytoplasm for translation. Second, they help protect the mRNA from being degraded by enzymes that would break it apart, so they help protect it from that. And then finally, once translation is occurring in the cytoplasm, these two modifications help the ribosomes attach to the 5' end of mRNA, and that's going to be crucial for translation. And these two modifications do these three things to help out your mRNA in eukaryotic cells. The other thing, so this is just kind of shows you, here's our 5' cap, so this is like a modified guanine, and then over here we've got lots and lots and lots of adenines. That's our poly A tail. That's added to the 3' end. So then we also, typically with RNA, end up getting in eukaryotes a long string of nucleotides, and many of which actually don't code for anything. There's a lot of like junk in there. And those non-coding regions are called introns. And the way you can remember that is that they are non-coding segments that lie between useful coding segments. So they are intervening sequences, hence introns. Okay. Then we also have exons, and those are the nucleotides that are going to be expressed, hence exons expressed as proteins. So those are the ones that are going to get translated into amino acids and then make us some proteins. So through a process known as RNA splicing, your mRNA will go through this processing to remove the introns and join exons together, and that way once the mRNA is transported to this to the cytoplasm, you now have an mRNA molecule with a continuous sequence of nucleotides that actually codes for something instead of having all of this junk. So you can see in this picture, we've got the bright orange represents segments that actually code for things. Those would be our exons. And our light orange is the non-coding segments, our introns. So this would be our pre-mRNA. Remember, it gets that 5' cap added, that poly A tail added, but then through additional processing known as RNA splicing, those introns are cut out and you end up with the final mRNA that's going to be exported out of the nucleus into the cytoplasm for translation, and all of it is coding. So that's very, very important. That is all for this section. So that's all you need to know, and very short and sweet.